The genetics of virus and bacteria. Viral DNA can become part of a host chromosome. A virus is an infected particle that consists of little morph genes in a box. It's a bit of nucleic acid wrapped in a protein coat. We call the protein coat a capsid, and then sometimes it has a little bit of membrane envelope around it as well. When a phage DNA enters a lytic cycle inside a bacteria, it is replicated and transcribed and translated. So that's what's happening here. We're in a lytic cycle. The phage injects its DNA, this light blue. The light blue phage DNA circulizes, and then that DNA is it's transcribed and translated, and new phage DNA and new phage proteins are synthesized. The new phages assemble themselves, and when the cell breaks open, the phages are released. We can also have a phage in a lysogenic cycle. And in this phage, the DNA is inserted into the host chromosome and can be passed on to daughter cells. So here we have our phage. He injects his DNA into the bacterial cell. The phage DNA circulizes and then incorporates itself into the bacteria's chromosome. So here it is, it's part of the bacterial chromosome. If the bacteria divides, that phage DNA is also replicated and ends up in both daughter cells. So you could have many, many cell divisions, and this phage DNA will sit there and not do anything. But then along comes some sort of environmental stress, and it'll go back to a lysogenic cycle, a lytic cycle. Right? Once it's inside, we call it a protophage. And its genes are pretty much inactive. All right. Many viruses cause diseases in plants and animals. Flu viruses and most plant viruses are RNA instead of DNA as their genetic material. And some animal viruses will steal a little bit of the cell membrane as they leave. It's going to help disguise them so that they can infect another cell. And mumps is an example of an organism that does this. It takes a little bit of the envelope of a cell that it has infected, and it helps it enter a new cell. And then I have a short video I want us to watch. This is viral reproduction cycle.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
So the last thing I've got is that emerging viruses threaten human health. Our book, of course, is pre-COVID, so it's not listed here. But you can see that viruses have been a problem for a long time. We've got the flu epidemic of 1918. A lot of people have compared our current flu outbreak with this one. There was a major problem not too long ago with H1N1. AIDS is caused by a virus. Ebola, avian flu, and these others. So we want to pay attention to our viruses, how they work.